So first of all, for those who may know me from meetings on the same around the same topic before the pandemic, probably as you can see from my current affiliation, I decided to switch roles after 15 years doing preclinical research and investing a lot of energy in building up predictive models, including MPS, to characterize drug candidates to enable entry into human testing. I changed roles and moved into clinical research, joining a team that is dealing with personalized healthcare approaches, and where we're also very interested to explore and see to what extent microphysiological systems could help improve clinical development and optimize the benefit-risk ratio for our patients and driving personalized medicine approaches. So that said, today you're not going to hear anything about predictive tools that I've been working on together with the team over the last years, and actually that's also not necessary because there were a couple of great examples during this meeting from my former colleagues, both on posters as well as presentations looking into this. Rather, what I would like to do today is share with you some of the ideas and goals and concepts that we're currently working on, and also give you some examples what we have been initiating studies. Clinical development has a bit of a longer time span than preclinical development, so those studies have not read out yet, so I'm not able to share with you like ready data and results, but I hope you find kind of the ideas and the concepts relevant and interesting. So how do we move this forward? Maybe here? No, here? How does this work? Now it works. Okay, so very generally, the basic kind of difference how we look at the problem, what problem we are dealing with. So a lot of the work that's being done with microphysiological system in the preclinical discovery space is around having models in place that are able to test quite large numbers sometimes of different types of drug candidates in order to select the best ones, those with the best properties, the most promising ones to move forward. And you typically do that with a handful of different cell systems next, of course, to other types of tools, which aim to recapitulate what I would say is like the average human. Now, when you look at the situation in clinical development, it's actually a bit a reciprocal approach, quite different, because here you typically have one single candidate where you invest into and which you want to move through the whole stages of preclinical development. But you need to be able to reflect the huge diversity of patients that you typically have. So once you have decided on a candidate, you don't want to stop that. You want to see, okay, which are the patients that are at risk, which are the patients that benefit most, and how can you find that out? And this is where we would like to introduce patient-specific organoids or organoids that represent a specific patient subpopulation. Can we use those to generate a patient-specific response and then aim for an individual treatment? So the graph that you can see here, I took that from a publication or like a leaflet around some basics of personalized medicine. So fortunately, it's not that dramatic that 80% is waste, but it can happen. But I would say our preclinical work is a bit more promising and successful. So it's certainly not 80%. But as you can see, then in an example that I will be giving just later on, we do have some areas where actually we're really struggling with relatively poor, relatively small fraction of patients that do respond well. So overall, what is our vision? So we really see MPS as a window into our patient's individual physiology for a more accurate prediction of patient safety. And we think this could nicely complement what's already there. So a lot is already being done in the personalized healthcare arena for clinical development. So looking into histopathology, there's a lot of genomic work being done. So we would like to combine that and thereby improve the patient experience. Minimize hospitalization is an important topic for us in clinical development. Of course, we would like to predict the response and identify those patients that would benefit most. So kind of both efficacy and safety aspects that we want to look at. We want to use MPS to develop tailored approaches to manage those toxicities. So rather than just kind of in the worst case giving up if you have severe safety signals or even fatalities, how can we predict it and then early on proactively manage those toxicities so that in the end a drug which may work for a large portion of the patient population can actually be given to patients who suffer from diseases. We also would like to use it to define optimal treatment regimens. So I'm particularly thinking there of combination approaches in oncology. To my knowledge, in our current portfolio, there's not a single agent where the intent is to use the drug as a single drug. So it's always combinations. How can we define the optimal combination approach? We think MPS can also really help us there. And ultimately also optimizing trial design. This is a bit of a stretch goal. I mean, there the data really have to look very, very good and promising. I would like to later on give you an example, like predicting the response. So there's one example that I will allude to just later on, predicting immune-related adverse events in trials. This is something that we're struggling with a lot in the immune oncology space. And also another example about how can we identify those patients at risk when we know already there seems to be some signals maybe in phase 1 studies or in early phase 2 trials. And we do see a couple of patients that shows some signals. What can we do by virtue of using microphysiological systems to develop mitigation strategies once we go into really heavy, large, very expensive trials? So very generally, a few minutes just on personalized medicine. So what is this actually? What are we doing here to get insights into patient-level drivers of response to improve outcomes and reduce the burden of healthcare on society? So this has a couple of different elements. First of all, trying to understand what are those factors that impact an individual response. A lot has been done already about genetic drivers. HLA typing is something that has almost become a bit of a standard as soon as you have something popping up there looking into this, and I will allude to that a little bit later on. Ancestry is something that's coming up more and more, and I was very pleased to see that actually also part of the MPS community is starting to think about that. Is quite tricky actually also, how do you define that? So ethnicity, ancestry, and the genetics underlying that, these are difficult terms to precisely address, but is absolutely a need, and is coming more and more also as a regulatory request that we look into that. Of course, also underlying diseases, age groups is again an important topic that I hopefully will be able to talk about later on. We have some projects in place using MPS to try and address different age groups, which again is technically a bit difficult, but we hope to be able to have data there soon. Of course, also in clinical development, it's a lot about prediction, biomarkers. Can we use MPS also to develop new biomarkers next to genetic testing? Monitored in patients, this is a topic a bit apart from MTS. Digital tools for remote monitoring to reduce hospitalization is an important element there, and also help physicians then for post-marketing in clinical practice. How can we guide them by providing them with additional information from our labs and from our investigations? So precision medicine then makes use of a couple of key enablers, key technologies, as alluded to for patient profiling. Genetic testing is currently being done at Roth for about half of the patients, I would say. So this is typically included, and most of the patients actually agree to the consent of being genetically tested. And on a routine basis, next to the normal clinical chemistry, things like cytokines, chemokines are being measured for patients. So this is an important aspect. Then in order to validate our systems, and this we need data. We need real-world data. This has been part from my preclinical presentations over all those years, access to patient samples, patient cells, but also the patient data. I still think this is something we haven't solved yet. It's not so easy to access this data. Tools like natural language processing is something that we currently apply to really leverage kind of historic data, patient records that are in PDF format at best or hard copies. How can we leverage that to have more clinical data that we can use to build those systems? Digital tools, I mentioned, this is a bit of a different topic. And what we would like to introduce now is a new kid on the block for clinical development and personalized medicine, patient-derived tissue models. So can we use these to in vitro to mechanistically understand and predict individual patient response? Now to a case example, cancer immunotherapy is an important part of our portfolio. When this whole thing started, it was believed that large molecules, they typically don't show lots of side effects, and we had to learn over the years, including also many other companies, that this is actually not at all the case. There's very promising results, sometimes really close to cure. However, as of today, we have to realize that only quite a limited number of patients actually respond to immune therapies overall. We have a relatively high rate of adverse events, and we don't have really good tools to predict them. And even more interestingly, and I will allude to that later on as one of our examples, that was really a starting point for our group. It has been observed by a couple of groups that about 70% of patients that had to be discontinued due to adverse events actually showed the best treatment response, hinting towards a link between the occurrence and the risk for adverse events in the immune oncology space and efficacy. And so we thought about that and did some prior investigations and are currently using that example to see can we do more with that for a personalized healthcare approach. So just very quickly here, so here's the whole range of clinical toxicities, going back to that statement that we heard earlier on when I worked at Roth also in preclinics. Yeah, antibody-based drugs, they're fine. They would not induce any clinical toxicities. This is actually not at all the case. It's quite wide here. So these are two famous prototypical checkpoint inhibitor drugs, and you can see like a wide range, both in grades and also types of organs addressed. So we do need tools to address that earlier on during stages of candidate characterization preclinically, but also those well-characterized molecules. Once we go into clinic, this is how it looks like. So we need better tools to manage that. So what has been done over the past years, including our team at Roche, we set out to build a suite of different types of models, typically consisting of primary human tissue, polarized where necessary, adding PBMCs in flow. All the tissue components are either donor matched in the best case, if possible, or at least HLA match. And so this is the basic simplified setup of those systems. That have been set up, and those systems then have been used to recreate the key processes that we're interested. So the drug-induced activation of immune cells, and then initiation of downstream events at the site of action, and there really have something that to measurable at the site of action in order to take decisions based on that. So in order to achieve that, kind of three main aspects needed to be solved for this example. Key event of immune cell activation. As you can see here, an example of the recruitment of leukocytes following stimulation of an endothelial system, then targeting the healthy tissue, as you can see here in the middle, These are hepatic spheroids, which have been treated with a PD-1 checkpoint inhibitor. And you can see the infiltration of leukocytes. And ultimately, in safety, we would like to see, so what's the consequence of that? Is anything happening? And here you can see, for example, this is classic chemotherapy-induced apoptosis of intestinal organoids. So that required over the years quite a bit of work to test out and benchmark these different models. And as said, you've seen now over the course of this conference some of the examples that have been shown. For example, the work Nicole Czeczorewski presented earlier on, where we did establish such models to use them to characterize and select the best molecules early on. Now, the same type of systems we do now also want to use in the clinical space, but it comes with additional requirements. So here, first, a bit of an outline, what kind of effects we're dealing with and where actually human tissue models can really be of help. Not everything can be solved with human tissue models. Cytokine release syndrome is still like a big problem. Somebody mentioned earlier the Ticinero case. That's quite some time ago. Whole blood cell assay has been set up as a consequence of that, and this is being done. Nevertheless, it has some limited predictive power, and still in clinic, it's something we see very often, and is still something that remains to be solved. Off-target effects is relatively rare for these type of molecules, and one can think of like peptide arrays and other things. Autoimmunity is a big issue there. We're very interested in T-cell repertoire sequencing that we do with patients. What I would like to focus on are so-called on-target off-tumor toxicities, because there you have already a bit of an idea based on the expression profile of your target, where it will hit, and this is where it has been used for example, for T-cell bispecifics, but also cell therapies, obviously. So this is where human tissue models in the preclinical space, but also in the clinical space, have been useful. Now back to the clinical example and what has really brought us to think about how can we make use of MPS for a personalized approach. This is one of these reports here, which shows us quite astonishing and also concerning data. So what you can see here, these are non-small cell lung cancer patients which have been treated with a checkpoint inhibitor. And the graph here shows survival and the probability of experiencing severe side effects. So different types of side effects, pneumonia, hypothyroidism, and so on and so forth. And what you can see here is a significant difference between patients that have immune-related adverse events versus patients that have no immune-related adverse events regarding survival. Yeah. So, of course, when a patient experiences immune-related adverse event, there's chances are that we have to take those patients off treatment if they really become severe. But as there seems to be this link between survival, we want to make use of MPS to try and address that. So how can we keep those patients on treatment while at the same time managing the toxicity rather than just taking them off treatment? So we first had a look into our own trials without going into all details. So these are just two examples of clinical trials with a Roth checkpoint inhibitor called atezolizumab. These are these two colored bars, immune-related adverse events in two trials that we ran at Roche. So you see quite a high incidence of adverse events, almost, it's a bit more than 30%, most prominently skin, but also GI and endocrine. And actually, as you can see on the right-hand side, we could confirm that the occurrence of immune-related adverse event was linked to better survival. So the team went out and analyzed those patient data, made a lot of genetic work and compared these values, the genetic data with the UK Biobank genetic resource, where they were looking for predictive genes that are associated with the risk for autoimmune type of diseases. So in this case, the examples were taken for autoimmune skin type of diseases like psoriasis or vitiligo. And so they built what is called a polygenic risk score. So the combination of different genes that pose, that can explain a risk for developing these type of autoimmune diseases. And actually, they could see that a higher polygenic risk score, yes, it does correlate with autoimmune type of, in this case, skin toxicity. And as you can see on the right-hand side, this higher polygenic risk also correlates with longer survival. So people that are on the treatment for cancer and at the same time are carriers of risk genes for autoimmune type of diseases, they are more likely to experience adverse events, severe adverse events, but they also have higher chances for survival. So this has all been published by our team in PNAS some years ago. And that was really the starting point for one of the projects that we are now kicking off. So we thought about, okay, if this is true, so can we use patient cells before they are treated? So as they are enrolled in a trial to be able to predict if there's a risk for immune-related adverse events. And thereby, in a proactive manner, implement early on mitigation strategies and at the same time addressing the tox, so while maintaining higher efficacy. So can we really solve that directly? So when you think of that, so if you want to test patients which are already in a clinical setting, so then you need to think of other aspects that become key and important next to the scientific criteria for, in this case, an organoid, a patient-derived organoid, whether you believe the data it generates is true or not. You also have to deal with minimal starting material. So you cannot just take like large amounts of biopsies, and patients will not agree to that. It has to be quite a straightforward, easy workflow. It has to be small, scalable approach that fits into a typically decentralized clinical trial setting. So when we think of a phase 2 trial, typically such a trial is not taking place in like one clinical research center, but we have patients decentralized over several places, because sometimes it's really difficult to recruit those patients. So you need to think of, okay, how do you organize all the logistics so that in the end you get fresh samples and you can process them into microphysiological systems. Furthermore, of course, as the clinical trial will not wait, the clinical trial will move forward. So it's important that the whole process is very short. So from biopsy to assay ready results where you can take decisions on. So this is not feasible if we're talking about several months there. So it really has to be short. Last but least, you need to bring that into the clinical protocol early on. And for, of course, the informed consent, which you may or may not get. So for some of these biopsies, we're talking about skin or also colonoscopies. So we're talking about roughly 50% or so of patients that would agree to such things. So you need to factor all of that in next to the tissue model that has to work, which is sometimes quite a bit of a challenge, and this is what we're currently working on. We do that together with a team from North Carolina. This is a startup called Silis. They have established a machine that is relatively small and is able to generate microphysiological systems, organoid type of very, very small mini organoids within 7 to 9 days. And we do that with different types of organs and would like to see, so how well do now drug treatment data match with a patient outcome? So these are studies that are currently ongoing, and I hope to be able to report back maybe next time we meet here. So this is all in the making. The goal of this is, of course, that ultimately, if it's successful, that if the patients enrolled in a trial can all be proactively tested, and then if we see that the organoid yields in relevant results, that we can define individual treatment and also for safety define individual measures. The whole idea here is that when we look now at how personalized medicine works in a clinical setting in oncology, so this is from the MD Anderson Cancer Center. So what we would like to achieve is something like the molecular tumor board. So this is something that has been introduced over the last years. So this is a series of about 20, 30 mutations that are routinely assessed for every cancer biopsy, which is then part of the treatment decision for a personalized approach. So these are the targeted therapies. And as you can see, we're actually well advised, or physicians are well advised to make use of that. This is from a recent review showing that actually a physician, if the team follows only the physician's recommendation for treatment, actually the chances for survival of the patients is significantly lower as compared to what the molecular tumor board is proposing. So if we're successful here, we would like to have a seat on that table and add patient organoid data so that this then in kind of a holistic assessment of the individual patient can contribute to more tailored approaches. So this is what's currently ongoing. Now for the last minutes, I would just like to switch gears very quickly for another very quick example around genetically defined systems. So as some of you may know, HLA types, they very often predispose for certain drug-specific adverse events, which also have a regional pattern of distribution. So on the left-hand side here, you can see some of the most famous examples. Abacavir is probably the most famous one, leading to hypersensitivity reactions in such a severe manner that it's actually required to test patients for that particular HLA type before a decision can be made whether the patient receives the drug. This is also something which has been found in association with other types of treatments, like here for Crohn's patients that receive these types of drugs. There's an HLA association with the development of anti-drug antibodies. So we had kind of a similar problem here. This is data from a phase 2 trial where we saw a number of patients that showed ALT elevation. So no severe DILI, otherwise the program would have been stopped. But quite a high number of ALT elevations, and pharmacogenetic analysis revealed that it looks like there's an association with a particular HLA type. And also on top of that, as you can appreciate from the graph there on the left, this HLA type is associated also with a certain ethnicity. So we asked ourselves, can we recapitulate that? Can we model that in an in vitro system to see whether actually, is it confirmed, first of all, an increased risk? Can we better understand it? And can we take measures? Because this program is moving forward into phase 3. Longer treatment, and also patients that come with significant comorbidities. So if we have already a risk factor, and for example, then patients have already, for example, inflamed livers, there's a risk of potential high-low type of severe DILI. So this is what we want to avoid. So we would like to use organoids to identify those patients early on. And we do that together as part of a collaboration with Custom in Cincinnati. They have established a very nice system which allows us to generate those genetically defined liver IPS models, which we're currently working on, where we hope that this will help us to initiate closer monitoring, maybe think of co-medications, altered dosing, or in the worst case, excluding patients from trial. So this is what I wanted to share with you. This is the overall concept. In summary, we're interested in looking both efficacy and safety at the same time to generate kind of an in vitro therapeutic window, use that for clinical decision making, assessing that window, defining optimal combination therapies, identifying patients at risk, identify patients that will benefit most, not forgetting that, optimizing trial design, and hopefully also reduce our development cycles. Some of these, for those who are interested, some of these thoughts and concepts have been recently published together with colleagues from Novartis, bringing together the whole genetic field with patient-specific microphysiological fields, use that for patient stratification and aiming for individual treatment. And with that, I come to an end. Would like to thank those people, both from preclinics and clinics, that are working with me on some of those projects, as well as some of the collaborators. Thank you very much.